Welcome back, dear students. This is Dr. Hadi here. Today we will discuss the photoreceptor transduction cascade in detail. Before this, on the same whiteboard, I have already discussed the visual cycle or the word visual cycle in detail. I hope you watched that video lecture and now you are with me in this lecture. In today's lecture, we will discuss how the light is converted into an electrical signal inside our eye. So the whole case will be discussed inside the eye, inside your eye. So here we have a light enters in our eye. How this light convert into current or nerve impulse will be discussed the topic. So let's go straight into the topic. This is your eye. The person is in front of your eye. The light enters your retina, and in retina small current is produced. It's fine. I will take out this retina from your eye and will place it here, and I will let you show that inside the retina, this is the retina, inside the retina there are number of cells. These cells are much in number, very high in number. Okay, so I'm going to write only 2 cells out of all these cells. One is called the rod cell, another one is called cone cell, and you should know that there are number of such rod cells and cone cell in your retina. Cone cells are involved in color perception. Means if you see this is a black color, cap is black color, so this is because of cone cells, and if it is white, this is because of cone, red, blue, and so on. The rod cell actually involved in perceiving the seeing objects in the daytime and even in the dark. So in the dark and in daytime, which cell help us seeing objects is the rod cell. So I am going to take out this rod cell and zoom it. I have written it here. This rod cell, same rod cell is there. The mechanism that lies behind the rod cell is same for the cone cell as well. So we are not discuss cone cell, only the rod cell will be discussed. Okay, before you take out the rod cell, if you look inside the rod cell, you will see that there is a very special molecule, conjugated molecule, that we have already discussed. It is rhodopsin. Rhodopsin is present here inside your rod cell. So this is our rhodopsin. In order to explain the lecture, explain the idea, I will take out this rhodopsin here. The whole rod cell is now in front of you. This is the whole rod cell. Let me write it, rod cell. So inside the rod cell there are some molecules, some receptor, some enzyme that you need to know. The very first thing, rhodopsin, is present on the membrane of the rod cell. So this is your rod cell, and the rod cell there is rhodopsin. So this is the very first thing. When we say light enters your eye and light falls on the retina, actually light falls on this rhodopsin. Okay, so here finally we got our place. This is our rhodopsin. And beside this rhodopsin, what other molecules we have? There is one more molecule called the transducin T. Transducin T. Normally it is in rest, it is inactive. Another molecule is the enzyme called phosphodiesterase. pHO phosphodiesterase enzyme. So here PD is the enzyme, and here is the another enzyme called guanylyl cyclase. Let me make it much simpler. This is another enzyme called cyclase. Okay, and we have some channels, small pipe-like structure through which ions can enter and exit. So these are called sodium channels. Sodium channels. Okay, so these are channels. So what we have in a single rod cell is the rhodopsin, of course, the most important thing, rhodopsin, and we have transduction, we have phosphodiesterase enzyme, we have cyclase, guanylyl cyclase, and we have channels. These 4-5 objects are there. Okay, what happens in the dark first? So before I start with how current is produced, let me shift to the resting time when there is no light. But means what happens inside your eye when there is no light. Okay, when there is no light, then there is a molecule called cyclic GMP, cyclic guanosine monophosphate. As you have heard, AMP, adenosine monophosphate, ATP, adenosine triphosphate. This structure of ATP have already been discussed in my video lectures. One of my video lecture, you can go and watch this. Just like ATP is a molecule, GMP is also another molecule. Looks like it is the cousin of ATP. So it is a guanosine monophosphate. But this guanosine monophosphate is having a cyclic form. That is why it is called a cyclic guanosine monophosphate. Cyclic guanosine monophosphate. It is present inside the rod cell and in a large amount. Which enzyme is producing the GMP? That the cyclase enzyme, this is the cyclase enzyme that is converting simple or you can say that this cyclase enzyme is producing cyclic GMP in simple words. So you got this. Okay. Now, when there is cyclic guanosine monophosphate in the dark, their level will be increased. Means as far as light is not there, guanosine monophosphate will be continuously synthesized inside the cell. So as a result of no light, we will be having a large number of cyclic guanosine monophosphate. The cyclic guanine monophosphate are the guards standing at the gate or the door of some institute. So they allow this door to open, this channel to open, so that sodium from outside enters inside. Means sodium plus. Here sodium from outside enters your cell. So as a result, we will be having large number of sodium ions inside your cell. Which cell? Rod cell. Because of what? Because of increased cyclic guanosine monophosphate. This increased guanosine monophosphate leading to the large number of sodium ions or positive ion inside the cell. These ions will make our cell what? Depolarized. Depolarized. In this state, what happens here? You can see a knob-like structure. Here there are small sac-like structure where another special neurotransmitter is present. Just like in case of acetylcholine, which are present in sac-like structure vesicles, here we have glutamate. We have glutamic acid, like an amino acid. So these glutamates will be released in a very large amount. So here we have glutamate in a very high amount. Large number of glutamate is there. This high amount glutamate, more glutamate will cause this cell. This is another cell. Okay, below the rod cell, here below the rod cell there is another cell. There is another cell called bipolar neuron. And after that bipolar neuron there is another cell called ganglion. And after this ganglion there is optic nerve. Similar is the case. After the rod cell there is bipolar neuron. This is bipolar neuron. And then we have ganglion. This is ganglion. And then we have the optic nerve. This optic nerve is leading to the brain. So this is the idea. Okay, once we saw a large number of glutamate is there, the cell seems to be on here. The cell seems to be on because of large number of sodium ions and large number of glutamate. This cell is on, but this caused this neuron to stop, off. This neuron is off, means okay, no signal will be there. Remember that more glutamate will inhibit this neuron. When glutamate is there, large number of glutamate will inhibit this neuron. So when no current is in that neuron, no current will be that, and no current to the brain. So no light, no current. No light, no current. This is the game. This is on and this is off. Simple. Now when the light falls on your eye, what happens? This cell become off. This cell become off and this neuron become on. How? Let me explain this. And this is most interesting part of the lecture that I am actually trying to explain, to describe this whole. Okay, now I will start from what? From the light. When the light falls on the rhodopsin, you know, is a vitamin A and opsin protein. This will break down into segments. This is of course the vitamin A segment. This is your vitamin A segment and this is your opsin protein. This is your opsin protein. When the light falls, what happened is already been discussed here. When the light falls, what happened? This opsin protein, it is actually got one another name as well. When you come to this pathway, there is another name called meta-rhodopsin. Opsin has another name as well called meta-rhodopsin. So meta-rhodopsin will remove a small segment from itself. This is a very small segment from the this one, meta-rhodopsin. A small segment, it will detach and it will come and attach here with transduction. It looks like that light has activated this structure. Means this structure, when it was present here in the conjugated film, it was inactive. It could not work. So what happened? When the light fall on this structure, slight changes took place, and because of these changes, the meta-rhodopsin is the active form now. This active form will release a small segment of it and it will attach itself to the transducin. Transducin is another molecule. Transducin was inactive. Now it is active. So transducin is now activated. This transducin will now come to an enzyme called PDE. Okay, this PDE Let me attach this transducin. Transducin is attached to the PDE. PDE was also inactive, but once the transducin is attached, it becomes also active. So I'm writing these lines just to make sure that this enzyme is now activated. Once this enzyme is activated, diesterase, it will start killing, it will start decomposing these cyclic GMP. So as a result of this enzyme, the cyclic GMP level will be decreased. Cyclic guanosine monophosphate decreased because of PDE enzyme, because of transducin, because of segment removal, because of separation of these two, because of light. So the whole cause behind everything is the light. Okay, so the decreased guanosine monophosphate will lead to the blockage of these channels. These channels will be blocked. When these channels are blocked, what do you think? There will be very less sodium ion. So less sodium ion will give us another condition called hyperpolarization. Hyperpolarization. If you remember, in the previous case, it was called depolarization. When the light was absent, it was called depolarization. But when the light is there, the cell become hyperpolarized. In hyperpolarized state, the glutamate will be not in excess. Very little glutamate will be there. Very little. More? So very little. Less amount of glutamate will be there. And this stage, we say that the rod cell is now off. It is the glutamate that tell us that the cell is off or on. So when the glutamate is very less, the cell is off. When the glutamate is high, the cell is on. And we know very well that the next cell after the rod cell is always doing opposite. Means if this is off, so this cell will become on. So when the small amount of glutamate is there, this cause to attach at some receptors over here, it will lead to the generation of positive ion inside the cell, and here a nerve impulse, a small current will be generated that will move from the bipolar neuron to the ganglion, and from the ganglion into the optic nerve, and finally into the brain, where the perception is received. Means what you see, you can see now. So this was all the case. Now I have mentioned here something. When the light is off, when there is no light, what happened? There is more cyclic guanosine monophosphate. There is sodium channel open. Sodium channels are open. More sodium come inside the cell. The cell is depolarized. This is called on. We say the cell is on. Then as a result of depolarized cell, more glutamate is released, and more glutamate cause the cell to on and the bipolar cell to off. No current, no impulse to brain. But when light is there, when there is light, there will be less cGMP, channels open, channel close, more sodium, less sodium, depolarize, hyperpolarize. Now you can go in reverse order. Hyperpolarize, cell is on, the rod cell is off, more glutamate, less glutamate is released, the cell is off, bipolar neuron is on, no current is there, impulse is there, and it will go to the brain. So we have discussed all these things in just one single video lecture. I would like to repeat once again. In a nutshell, what happened, that inside the retina we have two types of cells, the rod cell and the cone cell. Cone cells are involved in color, rod cells are involved in just vision in the dark and in the daytime, both. So taking out the rod cell, and we saw that inside the rod cell there is one special conjugated molecule called rhodopsin, and there is another molecule called transduction, which is in inactive form, and there is an enzyme called PDE phosphodiesterase, and there is a cyclase, and there is sodium channel. So when light falls and that it broken down the main conjugated molecule rhodopsin, broken down into two pieces, vitamin A and opsin. Opsin is also called metarhodopsin. Because of these changes, a small molecule will be released from the metarhodopsin and it will come to the transducin. Transducin was inactive, it becomes active now. Once the transducin T is activated, it will go to another enzyme, phosphodiesterase, attach with it and activate this enzyme. Once this enzyme is activated, it will cause a decrease in cyclic guanosine monophosphate. The cyclic guanosine monophosphate decrease level cause the sodium channel to close. Sodium channel close, sodium ion will no more come in inside. As a result of the inside become hyperpolarized. Hyperpolarized, very less glutamate will release out. Less glutamate will cause the bipolar neuron to on and generate the electric signal. So the whole thing that is responsible behind this is the cyclic guanosine monophosphate. If its level is high, no signal. If its level is less, there will be a signal. And the guanosine monophosphate level depend upon the light. When there is no light, guanosine monophosphate high. When there is light, guanosine monophosphate decreased. And everything is then responsible. What was responsible for this is this rhodopsin. And we know that inside rhodopsin there is vitamin A. So from this lecture we have realized that vitamin A has a very important role in the vision. I hope you got this video. See you in the next video. Please give us likes, share, and plus what? Give us praise. Thank you. Bye bye.